Hi everyone, I'm Brett Drummond. I'm the science communicator and co-founder of MStranslate, and I'm here again to talk you through another one of our MStranslate research summary videos. So let's get to it. All right, so the research that I want to talk to you about today has got to do with constipation and multiple sclerosis, and this is the research paper in question. So it was published very recently with the title Constipation-Induced Gut Microbiota Dysbiosis Exacerbates Experimental Autoimmune Encephalomyelitis in C57 BL6 Mice. Published out of China. Now that title is obviously quite a mouthful, but we're going to break down really simply what they've shown in this research and why it might be important for people living with multiple sclerosis. So I guess the first question that we want to ask is, why are we looking at research into constipation? Well, firstly, we know that it's a really common symptom experienced by people living with multiple sclerosis. And recent research has also shown that constipation can cause alterations in the gut microbiome. I guess this is of particular interest because we now have quite a lot of evidence to suggest that there is a potential role for the gut microbiome in the development of multiple sclerosis. So for all of these reasons, these researchers have gone to see what's the actual effect of constipation on the gut microbiome in an animal model of multiple sclerosis. And so if we think about what they've done here, as I said, this was done in an animal model using mice and looking at really two main factors, and I'm going to break down what they are. So they had mice that they induced constipation in, and then they compared those mice with normal mice and checked what happened to their gut microbiome. Were there any differences purely based on one set of mice having constipation and the other not? They then also went on to do further experiments where they looked at what happens in these mice with constipation if we then give them EAE, the experimental animal model of multiple sclerosis. What does that do to the disease severity? So if we focus on that first question, what happened to the gut microbiome in mice with constipation versus healthy mice, what they saw was that there were a number of changes to the gut microbiome caused by the constipation. Now, these changes were in terms of changes in diversity of certain microbiome organisms. Some species were increased, other species were decreased. Now, there's some quite interesting things in here in terms of the types of species that were changed, and I won't go into that detail in this video, but if you do want to know more about that, feel free to comment in the comment section below, and I would be happy to provide more information about that, because some of these changes were related to species that we have seen before in other studies about multiple sclerosis. They also saw some other things in here in terms of increased permeability of the gut in the mice with constipation, as well as increased blood-brain barrier permeability. So both interesting things that have been sort of implicated before in multiple sclerosis. But again, it's important to remember that in this experiment, they weren't looking at the animal model of MS at all. This was just mice with constipation versus mice that were healthy. And so then they wanted to go on and look at what happens when these mice then do have the disease. And so this paper is freely accessible online at the moment. So what's good about that is it means I can share some of the figures and the data and talk you through what they saw. So if we look at this figure on the right here, we can see that we have three different curves. And let's focus on each of these curves in isolation to begin with, and we'll explain what this is sort of showing. Here we have the gray curve, and this gray curve is just mice that have experimental autoimmune encephalomyelitis, or EAE, the animal model of multiple sclerosis. And so these were just normal mice that they've induced disease in. We then have two other curves that you can see here. We have the black curve and we have the red curve. Now, the black curve is mice that had constipation, that then they induced EAE in, and the red is mice that underwent a fecal microbial transfer using feces from the constipated mice. When they were able to pass feces, they took those, transferred them into mice that didn't have a gut microbiome and let the gut microbiome develop from that, and then they induced EAE, okay? So they have a couple of different experiments that they're looking at here. And really there's a couple of takeaway messages from this that we need to focus on. Firstly, we can see that in the mice with constipation, or in the mice that had the gut microbiome from these constipated mice, the black and the red curves, we can see that the disease starts earlier. So we can see that the disease started at about day 10 in these mice, as opposed to day 13 in the other mice. So we're getting disease coming on about 3 days earlier. So we have earlier onset. We can also see that the disease tends to be a bit worse. They had a higher clinical score, so more disease severity. And that was common across all of these points up until a maximum here at around day 25, which we can see is about the same in all mice, but it's worse in those constipated mice. So it's showing that constipation seems to lead to an earlier disease onset and a more severe disease outcome. Also, we can see that those changes appear to be due to the gut microbiome changes in these mice, and that's shown by that red curve here. So they wanted to know why that might be. And so here we're looking at the effects on the immune cells in these mice. Now, I'm not going to go into this in too much detail, and this is actually one figure that I've just taken a subsection on. So this is a bigger figure, but just looking at two different types of cells here. So we have Treg cells, which you can see here on the left. Now, Treg cells, or regulatory T cells, are the cells that are meant to keep the immune system in check. And we know that this tends to be impacted in people living with MS. They tend to have lower T regulatory cells than healthy individuals. And so in these mice, again, using the same colors, here we can see these are just the normal mice that have EAE. And here we have our constipated mice in the black and our mice with that same gut microbiome in the red. And we can see that both of these cases have lower levels of these regulatory T cells. They also looked at Th17 cells. Now, these are a type of T cells that have been impacted, that have been shown to cause some damage or seem to be involved in causing damage in multiple sclerosis. And so here we can see that the number of these cells, the cells that are causing damage, are higher in our constipated mice and in our mice with that same gut microbiome than in our normal control mice here. So we can see that there seems to be impacts on the immune system, both in terms of lower numbers of these regulatory cells, the ones that are meant to keep everything happy and in check, and higher numbers of those ones that might be causing problems. So that seems to show why we might have been seeing that earlier onset of disease and more disease severity in the constipated mice. So I think these findings are really interesting and tell us something new in terms of how constipation may also be involved in the onset of disease. Obviously, one question that we always have to ask in these sort of studies is, will this translate between an animal model of multiple sclerosis into seeing the same sort of findings if we looked in terms of people living with multiple sclerosis? And at the moment, we don't have the answer to that. But it's interesting to see how a really common symptom of multiple sclerosis may actually be involving maybe influencing the disease process as well. So as always, if you do have any questions on any of the research that I've talked about in this video, please do comment below and I'll make sure that I respond to all of the questions that we receive. And depending on what platform that you're watching this on, please do subscribe or follow MStranslate across all of our channels so that you can stay up to date with all of the latest research into multiple sclerosis that we're talking about. Thanks, everyone.